I'm the science guy, so you're going to be in science class now. No spitballs, okay? And if you're chewing gum, Jim, I say, do you have enough for the whole class? Okay, so you'll notice that we keep the troublemakers up front. So we've got Jim and Mike and Elaine up front. These are the troublemakers. I've got good peripheral vision, so Bill, don't try anything, okay? So we're going to talk science. And I want to give you two perspectives, one 25 years ago and one a little more recent. A personal perspective 25 years ago or so when I first started doing work in myeloma in a laboratory, I happened to go to a first grade class for my daughter. It was career day, and I was the scientist. And so I got in front of this group of little kids, and I tried to explain what a scientist does to first graders. And I said, well, a scientist asks how, what, where, why kind of questions and tries to answer them. For example, what would happen if you put one of your mother's plants in a dark closet? And the little boy raised his hand, and he said, she'd spank me. Perspective is know your audience, okay? The second perspective is that I often would introduce to audiences that myeloma is a disease of the elderly. Last year, I turned 60, and I realized that myeloma really is a disease of the upper middle age now, okay? So those are two more personal perspectives. So the problem in myeloma treatments, is, as you heard, is that we're all different. Look around the room. You all look different. So why would you expect that what's going on inside your body should be the same as the person sitting next to you? So we already know that there's a lot of differences among patients. Some respond to therapy, some don't respond to therapy. Some people relapse, and there's this enormous amount of heterogeneity in individuals and in their diseases. So why is it hard to treat? Well, it's heterogeneous. That is, not all myelomas are alike. If I want to travel from here to Chicago, there are a lot of ways to get there. And so there are a lot of ways in which cancer cells develop and are developing uncontrolled growth and go places they shouldn't go. And we need to try to understand that. Myelomas can evolve. So the myeloma that is diagnosed oftentimes becomes resistant to a therapy that it responded to, and it tends to evolve, literally an evolution of genetics that is evolving in that tumor. We need to understand that. I tell you that the myeloma of one patient is not the same as the myeloma of another patient. It gets even more complicated because even within one person's myeloma, there are oftentimes substructures of cells that are different. And some of the cells will respond to the therapy and other cells don't. And in true survival of the fittest, if 90% of your cells respond to the therapy but 10% don't, the survival cells will be selected and they will grow out. So we need to understand that. And relapse is definitely a problem. So the big question is, what makes every individual unique? Your genetics. You'll be tested on this, so you got to write that down, okay? So anything in red is something that I'm saying is important. Sometimes I put an exclamation point. Sometimes it's all in capitals, so I'm shouting at you, right? So whatever makes everybody individual is your genetics. Not only you as an individual, but the tumor cells that are being produced are individual for each myeloma patient. So let's think about the cellular flow of information. This is the biology part. So in every cell you have a nucleus, and this is the command center of that cell. It's where your DNA is. It's the DNA that controls everything that's going on in that cell. That DNA is expressed as an RNA. It's a message that comes out of the nucleus, and the amount of RNA that's being produced is then translated into the protein. And the proteins in the cell are the factories that tell the cell to contract if you're a muscle cell and to think if you're a brain cell, okay? So this is a general phenomenon in every cell that the DNA controls the message that goes out to produce the proteins. The amount of proteins that are produced become important. The nature of those proteins are also important because what happens if the DNA has a change in it such that the factory that's being produced has fewer smokestacks? It doesn't work as well. Or what if the change in the DNA produces a factory that has too many smokestacks and it's working too well. That is, what if that factory is telling the cell to proliferate, to divide, to grow, and you've got too much of that? That's all happening at the level of genetics. It's all happening at the level of DNA, and we want to understand that. Because if we can understand the biology, we can understand where we can target the treatments. Okay, so this leads then to this whole idea of this human genome project, where we want to know the sequence of all of the DNA in your cells. And that's not a trivial task. It took 10 years and about $3 billion to do it the first time. But what did we learn? Well, we learned in a genome that it contains 3 billion of these bases, these A, G, C's, and T's, that are arranged in a particular order that is the code for that particular organism, for you. 3 billion of these are lined up, and in the first Genome Project, they sequenced the order of all 3 billion of those. And what we learned is that you and the person next to you are about 99% identical, 99.9% identical. Well, that 0.1% difference is still a million differences. So we want to understand where the differences are and how they contribute to your individuality, risk for disease. Once you have a disease, how do you respond to therapy? So every myeloma, that's part of the test there, every myeloma is unique in its genetic characteristics. So what are the technologies that we have available to do that? Well, we now have the ability to start asking questions like, how many genetic events cause myeloma? Are there common events that some people share? Are there unique events? Do they affect progression? Do they affect response? Do they change over time? And so we can learn a lot by understanding the DNA, the genetics of the individual, of the myeloma, of many myelomas, and start analyzing the biology based on our understanding of the genetics. So what have we learned in the last 5 or so years in terms of genetics? Well, we've learned that there's a lot of changes in the DNA sequence. Myeloma is viewed oftentimes as being genetically unstable, okay? You've got a lot of DNA damages and things happening that I'll talk about. The problem is so much of that DNA change, we talk about the fact that some of these changes are drivers. They drive the proliferation that causes the malignancy. But because a lot of these changes are occurring, some of them are just passengers. And we've got to figure out which are the ones that are just going for the ride and which ones are the important ones driving the malignancy. Some of them are common, some of them are rare. Some of the DNA changes are associated with high-risk disease. Some might be seen at relapse, but they weren't seen at the diagnosis. Myeloma genetics changes over time. Some of them might actually help you direct better choices for therapies. So the take-home lesson here is myeloma is kind of complicated. It's got a lot of genetic chaos that's going in it, and not everybody's myeloma is the same. So, of course, we have a lot more data analyzed, and it says here, your medical problems are more complicated than I thought. I'm going to refer you to another doctor who has more medical insurance. Okay? So let's talk a little bit about the landscape of myeloma genetics. Well, at the 30,000-foot level, we can look at chromosomes, and you heard a little bit about chromosomes. So there's a normal-looking chromosome. You literally can isolate these segments of DNA and take a picture of them. And that's a picture called a karyotype. And you'll notice that there's chromosome 1, 2, 3, 4, all the way to 22. X and Y are the sex chromosomes. And you get 2 of each, okay? You inherit 1 from mom and 1 from dad. That's how the whole sperm and egg get together, and they each contribute half of your genome. So 1 chromosome came from mom, 1 chromosome came from dad. We can also look at the DNA sequence and ask at the gene level, because we know there are about 17 to 20,000 genes we could be looking at that control lots of different cell functions. So we can look at genes. We can look at how much of the gene is expressed and what its impact is on the protein. So the question then is, can we look at chromosomal abnormalities? What are they? We can look at mutations in the DNA, changes in the DNA sequence that may affect the amount of protein. And we can look at the amount of the protein that's made or the amount of RNA which is going to dictate how much protein is made. That's the expression of the gene. The gene may have a functional contribution, but its level of expression is also going to be important. So, the chromosomal abnormalities in myeloma. Now let me start by saying, remember those immunoglobulin proteins that are part of your immune system? It turns out that these cells that make immunoglobulins, plasma cells, in order for them to make the immunoglobulins, there's a really interesting characteristic that those genes exist in separate parts of the chromosome, and they have to reassemble in order to make a normal, appropriate immunoglobulin for immune system. Chromosomes have to break and rejoin. That's a normal process that gives you the capability of mounting a diversity of responses to the immune insults that you may get over your lifetime. That's a good thing. So if you're an evolutionist, you say evolution has developed this really unique characteristic that I can break and rejoin these genes in different combinations to give me lots of diversity that I can fight all kinds of invaders that come into the body. That's a normal function. The problem is that because of the function of breaking and rejoining, occasionally the breaking and rejoining doesn't happen just at the immunoglobulin genes, it happens at other regions of the chromosome. And when they break and rejoin, some of those breaks and rejoins impart new functions to the cells. Proliferate, don't die, go places you shouldn't go. I've just defined malignancy, okay? And that's what something like this might look like. So there you have two chromosomes that normally will divide on their own, and then they'll go through that process. But because these cells have a normal process of breaking and rejoining gene segments in a chromosome, occasionally they'll break and they'll mistakenly rejoin across chromosomes. So now I have a piece of A on B and a piece of B on A, where the chromosomes have broken and rejoined in different combinations. And that deregulates the genes that are located right at those boundaries. And some of those can contribute to cancer. This is what's called a circus plot, and it simply says that this is an indication of the chromosomal breaks and rejoins that occur in myeloma. And the width of the bands is how often you see it. So some of them are more common than others. Here's that case of 1411. You've also got 414. So you can see that there are a number of different chromosomal abnormalities that have been identified in many different myelomas. Now that doesn't mean every myeloma has every one of those, but myelomas can be characterized on which of those kind of chromosomal rejoins is taking place. And this is just kind of a summary slide of the potential that has been seen in a variety of different myeloma cells. If you do a karyotype, there's our normal karyotype on the left. There's one copy of each, one from mom and one from dad. The other thing that can happen in myeloma is the myeloma cells get too many chromosomes. And we call that hyperdiploidy. Diploid being 2, hyperdiploid more than 2. And you can see in this particular karyotype, instead of 2 chromosome 1s, they've got 3. 3 in 3, 311, 315, 3 in 19. So in myeloma, it's not unusual to have multiple copies of entire chromosomes. And that can kind of play havoc in what genes are expressed in the cell as well. In addition, there are examples where there are pieces of chromosome missing. Where did they go? I don't know. When the chromosomes started dividing, pieces of them fell out of the cell. And here's one where an entire chromosome 22 is missing. They only got 1 copy. So this again is an example why there's a complication in myeloma to try to understand the biology, because look at what's happening in the genetics at the chromosome level. These chromosomes are breaking and rejoining, they're deleting, they're adding copies. This makes myeloma a bit complicated. Having said that, we know where to look, and people are defining these kind of characteristics as they're associated with individual myelomas. This is a busy slide. You will not be tested on this slide, so Bill, you don't have to take notes on this, okay? But it simply says that all of those cytogenetic chromosomal abnormalities have been identified in various myeloma patients, and over the years with accumulated data and understanding how each of those might impact the myeloma, there are some summaries that can be made that says, well, for this one here, the 1416, tends to be the high-risk myelomas. That makes myeloma a much higher risk. Some of them might be an intermediate risk, and there are some suggested therapies that might be associated with that. So this is the first indication I'll show you where understanding the genetics and the chromosomal abnormalities are starting to give us a better understanding of the biology associated with that and starting to direct what might be some appropriate therapeutic approaches. At the same time, individual genes within the genome can be mutated. Remember the factory that has too few smokestacks or too many smokestacks? Well, this is just a list, not being tested. This is a list of the names of genes out of 17,000 that you could look at, are the most common genes that are seen to be mutated, the DNA changes that change the structure in myeloma. And you can see the RAS genes are up there. What does RAS do? It causes cells to grow and proliferate. So too much RAS, in this case, highly activated RAS because it's mutated, is a bad thing because it's telling cells to proliferate. BRAF right there, that's common. It's also common in other malignancies. And in fact, there are now directed therapies against people who have BRAF mutations. As you heard earlier, we're now defining those mutations not associated with myeloma, but those mutations in genes that may be associated with other malignancies. And so now the pharma companies have much more incentive to use these therapies in a variety of malignancies that might share some of these common mutations. And then, of course, myeloma adds another complexity is it doesn't sit still. It's these bad actors like these guys up front. They're bad actors. And as time goes along, they add mutations. There's this clonal development of genetic instability so that what you're diagnosed with and what a patient may relapse with may have different characteristics. And so what I'm pushing here is that we really need to understand the genetics, not only at diagnosis, but at various stages of the disease. Some of these will influence the progression and how they respond. Genes are expressed at certain levels, and this is what's called a heat map. So let me explain this to you. What John Shaughnessy did a number of years ago is he looked at the level of expression of all 20,000 genes in myeloma cells and asked, if I compare myeloma cells to normal plasma cells, these are the malignant ones, these are the normal ones, can I identify genes that are deregulated and expressed at higher or lower levels that distinguish the malignancy from the normal cells? And so what the computer did is it took all of that data and it narrowed it down to 120 genes. There's 120 genes, and each row is a different gene. Each column is a different sample. So there are a lot of samples of normal plasma cells, a lot of samples of myeloma, and you should immediately see there's a pattern here. There's a pattern that says in normal plasma cells, red indicates high level of expression, green is low level expression. So myeloma is not simply genes that are expressed too high, it's a combination of genes that are expressed too high as well as genes that are expressed too low. Lots of red genes in normal plasma cells that aren't expressed in myeloma. Lots of green genes that are not expressed in normal plasma cells that are expressed in myeloma. So there's a real pattern here. But there's another important part of the pattern that I'm going to bring back. And that is, if you look at all of these myeloma cells, no two are exactly alike. There's a lot of green here where there's red in that myeloma. And so even though there's a common pattern to expression of genes, we also need to recognize that there are no two myelomas that have exactly the same expression pattern, because that might be important to understand the differences in therapeutic response. So genomics now has demonstrated, should have been in red, take notes, okay? Chromosomal changes happen, gene mutations happen, changes in gene expressions. How can we use that information to better define the right therapy for the right patient at the right time? So has myeloma changed in the past 10 years? No, it's still a complicated disease 10 years ago. We just understand the complication a lot better now. And so the good news is your cholesterol stayed the same, but we have a lot more research findings that those have changed. So that gives us better information. So let's get back to this and say, let's look a little more detailed at this expression kind of thing. And the fact of the matter is, I didn't name the genes here. I just gave you a pattern, right? These are malignant, those are normal. So there's a pattern recognition here. Where have you seen pattern recognition before? You see pattern recognitions in barcodes in the grocery store. So look at this pattern here. This pattern looks very similar on the right and the left at the lower part. They have exactly the same bars in their grocery store pattern. But right up there, right about up here, around the 5 and the 1, you can see there's some thick bars and thin bars that start changing. So what that would probably tell you at the grocery store is there's something that these two products share in common, but there's something that distinguishes them. One's white bread and one is wheat bread, okay? So they're both myelomas, okay, but one is different than the other, and we could tell that difference by the barcode. Imagine if you could take a gene expression pattern of a patient sample and identify from that a barcode reader that says you're going to have a good response to a drug or you're going to have a bad response to a drug based on your barcode on that expression pattern. That's what we've been working on, and that's also what John Shaughnessy developed this gene expression test to define high-risk disease based on the expression of 70 genes. So he developed 70 genes and he gave them a score based on how they were expressed. And depending on your score, he could define people that had high-risk disease, the red, people who die very early, or people who live a long time. So this became a diagnostic marker of high-risk disease. So let's look at a myeloma cell. So now what I'm going to do is I'm going to take that myeloma cell and I'm going to treat it with Velcade, okay? And this is a cytotoxic response. So here you can see the cells are dying as I increase the dose of the drug. That's a good response, okay? Now the question is, do all myelomas look like that? Based on what I told you, yes or no? Okay, no, good. Bill, you didn't have to take notes on that. The class got it right. Okay, so that's what it looks like. So we looked at 50 different myelomas, and by gosh, there's a lot of diversity in response there. Some cells die very quickly, and other cells don't die well at all, even when you reach concentrations of the drug that you could never even give a patient. This was done in the laboratory. So now you ask the question, I just told you that there was a way by expression to tell the difference between a myeloma and a normal cell. Can we do the same thing and say, what's the difference between the cells that respond really well versus the cells that don't respond? Let's do an expression analysis. So we're going to compare the expression profiles, and Dr. Mitra in our lab defined now another set of genes. Now he's got 42 genes. Again, every gene is a row, every sample is a column. What distinguishes those two patterns is the ones on the left came from cells that responded real well to the drug. The ones on the right came from myelomas that did not respond well to the drug. So imagine if somebody walks in the door and we look at the expression of just their myeloma, and it looks like that. Well, it's a barcode. Which barcode does that match up with? Well, it kind of matches up with the sensitive barcode. This would be a good candidate for Velcade, since that was done with Velcade, with a Velcade therapy. If the patient had this barcode look, it might be that that patient might not be expected to respond to Velcade. And actually, we've now tested this against clinical trials, and in clinical trials, the use of that defining expression distinction is pretty good in separating patients that do well and patients that do poorly on proteasome inhibitors. Let's take this one step further. Remember, we've got this class here, and all of you are good students, except Jim, Mike here, they're bad. They're the bad actors, right? Well, what if the myelomas have a population of cells that respond really well, but there are a couple of bad actors in there that don't respond well? Everything I showed you now is the bulk population. We now have the ability to look at this kind of expression analysis one cell at a time. So this new technology allows us to look at the expression of each individual cell in a tumor population and ask, is every cell the same? Differences between different patients, are there differences in different cells? This is a patient sample, and what Ahmed has done is he's created a probability of resistance score to proteasome inhibitors. And each one of those numbers is a score from one cell. And you can see there's a distribution of scores. There are some cells that are predicted to have low probability of resistance and some that have high probability of resistance, suggesting that that individual tumor has some good actors and some bad actors in it, okay? And then we've done that on a number of patient samples with the Mayo Clinic, and each one of these spots represents that probability of resistance score. Patients on the left, the three on the left, have most of their samples have a high probability of resistance. They did not do well. They did not respond well to the proteasome inhibitor. These three patients responded really well to the proteasome inhibitor. They had low probability of resistance in almost all of their cells. What about those two? These are two patients that have a bunch of cells that look like they're below the threshold that they should respond well, but they also have a collection of cells that are above the line that says, there may be a subpopulation there that we ought to take a look at. And we're in the process now of following up to see if this holds up. So, the notes, the take-home lesson. Myeloma genetics is really complicated. But we're using this complicated data to understand the variation among patients, the influence of genetics on disease progression, the influence on response and relapse to individual therapies, the variation even within one person's tumor population. We have technologies now that allow us to test that and evaluate that. So the ultimate goal is to have better predictions of outcome. It's one thing to say you have high-risk disease. It's another thing to say you have a disease that might benefit from treatment A versus treatment B. That's a different application. With better therapeutic choices is the goal. So I'm going to end and just say you do have to listen to your doctor. The science is continuing. We don't have all of this refined, but you have to listen to your doctor. And he says, yes, that was very loud, Mr. Trainer, but I said I wanted to hear your heart. Okay? Class dismissed. Okay?